DNA transcription is essential to life. It is the first step of many in the production of cellular proteins. A cell uses different regulatory mechanisms to vary the rate of gene transcription. It is the balance of these mechanisms that controls transcription and ensures cellular survival. Transcription is the process of creating a single strand of RNA from a double-stranded DNA template. Initiation of transcription requires several tartar box associated factors, the most important being tartar box binding protein, or TBP. Tartar box associated factors and TBP make up a complex called TFIID. TBP binds to the tartar box. This induces a 70-degree wedge in the molecule of DNA to make it easier for the enzyme RNA polymerase to bind to the promoter. Various activator proteins may also bind to specific sites along the DNA. The multi-protein complex mediator then interacts with the activator proteins to finalise the transcription complex. To release RNA polymerase, transcription factor TFIIH phosphorylates RNA polymerase to initiate transcription. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation. Within the chromosome, DNA is tightly packaged into chromatin. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of two helical turns of DNA wrapped around a core of 8 histones. The histones are involved in maintaining the nucleosome's shape and structure. Histones are acetylated by histone acetyltransferases, unwinding the chromatin in a conformation called euchromatin. This makes it easier for the transcription complex to bind. Histone deacetylase deacetylates histones, causing chromatin to wrap up tightly in a conformation known as heterochromatin. Some hormones are able to enter cells and regulate gene transcription directly. These are known as steroid hormones. Glucocorticoids are a major type of steroid hormone, and the principal hormone is cortisol. Cortisol controls expression of the genes associated with blood glucose regulation. When cortisol enters the cell by diffusion, the hormone then binds the glucocorticoid receptor in the cytosol. This causes heat shock protein 90 to dissociate, revealing a nuclear localization signal which was masked by heat shock protein 90. Then, the glucocorticoid receptor binds to an additional glucocorticoid receptor hormone complex to form a dimer. This then migrates into the nucleus. In the nucleus, the receptor binds to glucocorticoid receptor element, activating transcription of genes such as PEP carboxykinase or tyrosine aminotransferase, which control blood glucose levels. Genomic imprinting. In some genes, the maternal or paternal copy is specifically chosen over the other. An example of this is genomic imprinting of insulin-like growth factor 2, of which the paternal copy is specifically expressed in mammals. In the maternal chromosome, the protein CTCF binds to the insulator, blocking the effect of the enhancer. Whereas in the father, the DNA making up the insulator has been methylated, preventing the CTCF protein from binding, allowing the enhancer to activate transcription of IGF-2. RNA interference is the process whereby RNA molecules inhibit gene expression by neutralizing specific messenger RNA. MicroRNAs are derived from actual genes and begin as primary transcripts or pri-microRNA and are usually transcribed by RNA polymerase II in the nucleus. The pri-microRNA transcript folds into a stem loop structure. Then, DROSHA, a nuclear RNase enzyme, interacts with the specialized RNA binding protein DGCR8 to form the microprocessor complex. The complex cleaves the pre-microRNA transcript, producing what is known as the microRNA precursor. Pre-mRNA is then transported into the cytosol through exportin 5 for further processing. In the cytosol, the precursor is cleaved by dicer endoribonuclease. To produce a 21 to 24 base pair molecule of microRNA. The microRNA consists of a passenger and guide strand. MicroRNA then associates with the Argonaute protein that is dedicated to microRNA-mediated silencing reactions. The passenger strand is then released, and the resulting structure is called the RNA-induced silencing complex, or RISC. Gene silencing can then follow one of two routes. In plants, RISC can target specific messenger RNA strands thanks to the remaining guide strand and directly cleave messenger RNA with help from associated factors and ATP. In animals, less extensive matches between the guide strand and messenger RNA means RISC prevents gene expression through inhibition of translation. Both processes result in eventual degradation of the messenger RNA.